The overall goal of this procedure is to perform single-molecule analysis of the distribution of laser-localized interstrand crosslinks in genomic DNA in order to study cellular responses to DNA interstrand crosslinks.
This method can help answer key questions in the DNA repair field regarding the distribution of DNA lesions introduced by laser technology, a widely used experimental tool. The main advantage of this technique is that it allows direct imaging of DNA lesions introduced by the laser-activated compound in genomic DNA. This has not been possible before. Demonstrating the procedure will be Durga Gichimu and Gali from our lab.
Every time the targeting laser is used, it is important to perform a mirror slide calibration to verify that the laser is hitting at the appropriate XYZ coordinates, and to establish the area of the field captured by the camera where the laser can be set to strike. Find a field on the slide that has been previously etched. Set the exposure time and laser power, then adjust the focus to move to a nearby non-targeted field on the mirror slide. Start the calibration procedure. The software will fire the laser to etch the mirror slide along a horizontal and diagonal line. These lines define the boundaries of the field in which the laser can strike a target cell. Set several regions of interest, or ROIs, inside the field